So Dan, if we may have the first slide. I see we have 59 participants. That's impressive. So the first slide tells you what I'm going to tell you. This is going, as you can tell from the outline, going to be a deep dive into science. Rachel is worried that by the middle, your eyes will glaze over, but I'll try to keep you entertained as we go. We'll just see how that works. So first, I'm going to give you a brief course in virology and the evolutionary biology of the coronaviruses. We'll spend a good deal of time on the epidemiology of COVID-19, some of its clinical features, so you get a sense of what kind of illness this is, particularly among the hospitalized patients. And then we'll talk about treatment and vaccine development, and finally, what to expect for the future. So the next slide, first of all, starts very basically telling us what a virus is. First of all, at the very end of the 19th century, plant pathologists were working on the tobacco mosaic disease and were able to demonstrate that a transmissible agent caused the disease, transmissible from plant to plant, that could not be filtered out by filters that would exclude bacterial causes. So the very first viruses became known as filtrable agents. Eventually the term virus, which is an old Latin term meaning poison, was applied to these filtrable agents. The structure of a virus is simply a nucleic acid wrapped in a protein capsid with or without a lipid envelope. It has no ability to replicate or metabolize or do anything else on its own. It can only function inside the cell of a higher organism, which it then is programmed to take over and turn to its own purposes, which is to replicate itself and deliver its progeny to the environment to infect other cells. It consists of both structural proteins that hold the virus together and functional proteins that carry out these nefarious activities inside a cell. It has mechanisms to attach to cells, to penetrate those cells, to uncoat inside those cells, and to deploy the polymerase, the enzyme that will allow either its RNA or its DNA to be replicated. Next slide, please. So this is the way we classify viruses. And the first criterion, one of the earliest to be used, is their appearance under the electron microscope. And I had the privilege of spending time in the laboratory of Dr. Fred Murphy at CDC, a veterinary viral pathologist who was the world's expert on just this talent. And in fact, he sat on the International Committee on the Taxonomy of Viruses and contributed to the scheme of classification that we now have, and which was in evolution when I was in Fred's lab. Fred was the one who first described the Lassa fever virus and also Ebola virus. These viruses are either RNA or DNA viruses, so that separates them from the very beginning. If they're RNA viruses, they're either positive sense or negative sense RNA viruses. The positive sense viruses are able to translate their code directly into proteins. The negative sense RNA viruses have to make a copy of themselves that is then positive sense before proteins can be translated from the codon. The genome may be either double or single-stranded. Viruses are either enveloped or not enveloped. The enveloped viruses tend to be more fragile, that is, to be inactivated more readily in the environment outside of cells. Now, you've already heard that the coronaviruses can last anywhere from 24 to 72 hours on surfaces. So that doesn't sound very fragile, of course, but that's in contrast to unenveloped viruses like poliovirus, which can survive in sewage and water for months, if not years. And then their capsid symmetry, that is the protein shell that surrounds and protects the nucleic acid, can be either helical or icosahedral. Next slide, please. So this is how coronaviruses are classified. First of all, they're positive sense RNA viruses, so they translate their proteins directly. They are enveloped, as you've heard. They're spherical with this characteristic club-like spike, or a series of spikes on their surface that give them that crown-like appearance, which you saw in the first slide, actually. They have a large non-segmented RNA genome. The influenza virus, for example, is segmented and has something like 9 to 11 segments of the DNA that are separate from each other and facilitate rapid mutation. There are 4 structural proteins: the spike protein that you've heard about, which is the protein that allows attachment to special receptors on human cells called ACE2 receptors. That stands for angiotensin converting enzyme receptor 2. Angiotensin converting enzymes, very important in the regulation of blood pressure and a number of other functions. And human lung cells, particularly pneumocytes type 2, and gastrointestinal cells, a few cells scattered other parts of the body, are very rich in these ACE2 receptors that are out there to receive the coronavirus. Now, that's not true of all coronaviruses, but it is true of this one and the SARS coronavirus. Then there's an envelope protein and a matrix protein, which help to pull together the envelope, and the nucleocapsid proteins, which cover the nucleic acid and protect it. And then there's some 15 non-structural proteins, which are functional proteins and include that polymerase that I referred to, that is the enzyme that allows replication of the viral RNA. Next slide, please. So here's a picture of the virus. You can see those club-like spike proteins all around the surface. Within each spike protein is a receptor binding domain that directly attaches to the ACE2 receptor, which you see at the bottom of the slide. You can see those sort of studs over that coiled RNA in the center. Those studs are the nucleocapsid protein. And then you can also see the envelope protein and the membrane or matrix protein embedded in the wall or envelope and capsid of the virus. Next slide, please. So here is the replication cycle of the virus. On the upper left, you can see the coronavirus attaching to an ACE2 receptor. Then you can see the cell membrane invaginating to receive the virus. That's called endocytosis. And then inside the cell, you can see that it uncoats. That's a special process that some of these functional proteins facilitate. And then the genome is released. It is then replicated with the help of that RNA polymerase that I referred to. And then each one of those multiple strands begins to translate the proteins. The nucleocapsid protein accumulates in the cytoplasm, and whenever it finds an RNA strand, it sticks there. And then the membrane protein and the envelope protein arrange themselves along the endoplasmic reticulum of the cell, and then use that, as you can see on the right-hand side, to envelop these nucleocapsid-covered RNA strands floating in the cytoplasm. And then the virus exits by sort of a reverse endocytosis, or what we would call exocytosis. And then you can see all of the viruses that are accumulating outside the cell looking for more cells to infect. So that's the cycle. And each one of those points is a potential point of attack for drugs that might inhibit the infection with this virus. And it's the polymerase that we've attacked first, and we'll talk about that toward the end of the talk. The next slide shows the four genera of the coronavirus family. Notice only alpha coronaviruses and beta coronaviruses include those that infect humans. Now, we've known about 4 coronaviruses that infect humans routinely every year. They just cause the common cold. 229E and NL63 are alpha coronaviruses. And OC43, which Kenneth McIntosh at Harvard and later University of Colorado first isolated in the 1960s, and HKU for Hong Kong University 1 was also isolated around that time. All of these just caused common cold. And so most of us, except that we kind of thought the coronavirus morphology was interesting and cute. It really didn't pay very much attention to coronaviruses until 2003. We thought they were of no practical interest. They cause maybe 5% of upper respiratory tract infections in the winter. Under some years, some conditions, maybe up to 30%, but they're mostly of trivial interest. But in 2003, of course, SARS came along, caused by SARS CoV-2, a beta coronavirus, which is harbored by Rhinolophus bats. And in the SARS epidemic in 2003, was transmitted to humans through civet cats in these wet markets or live animal markets in China. That outbreak started in Guangdong. So I won't tell you about gamma or delta coronaviruses, except they exist. Next slide, please. I'm not sure I understand why we're getting that echo, but here is the Rhinolophus affinis, which is the Chinese rufous horseshoe bat. The horseshoe bats, that is these Rhinolophus genus of bats, are so named for the shape of their nose. I think you can, if you look closely, can make out the horseshoe shape of that bat nose. Next slide. And these are the pangolins, which are thought to have been the intermediate host between Rhinolophus bats or Rhinolophus affinis and humans in that wet market in Wuhan, pardon me. And in fact, that's based on recovery of an RNA sequence that was in pangolins that were recovered by Chinese customs being imported from Malaysia into China with the destination of those wet markets. Next slide, please. So Rachel thought I'd get rid of this slide, but is here just to sort of enumerate the variety of coronaviruses that are found in bats. And in the interest of time, next slide. These are other bat viruses, some 137 bat viruses. So bats are unique in being able to live with these viruses. The rabies virus is about the only one that actually makes the bats sick and even kills the bats. And it's part of a subfamily called lyssaviruses that are all rather similar to rabies. So all of these cause rabies-like illness, just not to the worldwide extent that rabies itself does. So the other bat viruses seem to live in harmony with the bats. And so there's considerable research on how that happens, why it doesn't make bats sick, but it makes us sick. Now, in this collection, I'm only going to talk about Nipah virus because I think it serves as a useful template for interspecies transmission. Nipah virus was first discovered in an outbreak of febrile illness and encephalitis in swine farmers in Malaya. Now, how did the swine farmers get it? Well, they got it from their swine. How did the swine get it? Well, this is a virus that's harbored in fruit bats. And fruit bats behave like the deer in our yard. That is, they come in, raid the fruit, they take a bite, and then drop the fruit to the ground. And that's where the swine consume the fruit with its contamination of bat saliva, and then acquire the Nipah virus and make the swine sick, and then the swine herd sick. This has been, in recent years, a source of major outbreaks of illness in Bangladesh. Next slide, please. So coronaviruses are not the only example of interspecies transfer from bats to other animals to humans. So let me take you back to 2003, when we first realized that coronaviruses might be important after all. Here is the epidemic curve beginning in November 1st, 2002. So this also, like our current pandemic, began in November. You'll notice, however, that it petered out over the course of 7 months, leaving us with 8,437 cases and 813 deaths, including a major outbreak in Toronto, especially affecting healthcare workers. But it was gone by July 11th. And there have only been a total of 7 cases of SARS since then, and all of them in China, 3 of them laboratory accidents, actually, and the very last one was in early 2004. So the great mystery is, where did SARS go? Next slide, please. So I'm showing you a little bit about the SARS epidemic just as kind of a template for the effect of air travel in the spread of both epidemics, SARS and SARS CoV-2 or COVID-19. So Dr. A, having treated a patient with pneumonia in Guangdong, then went to Hong Kong to visit family and stayed at the Metropole Hotel on the 9th floor. All of these other cases, A through or B through M, also stayed on the 9th floor. So presumably the transmission was down the hall, 2 other rooms, on aerosol. Notice that Dr. A went to a hospital along with H and J, and some 95 healthcare workers in Hong Kong hospitals became infected, and some more than 100 other close contacts of these 3 patients and the healthcare workers themselves. B went to Vietnam, where there was a major epidemic, including 37 healthcare workers, and a famous WHO epidemiologist who went to Vietnam to study the SARS outbreak there. And that was Carlo Urbani, who died of SARS. No, 21 other close contacts. CD&E went to Singapore, where there's another outbreak, including 34 healthcare workers. ILM went to the United States. Only one healthcare worker in the US was affected. None in Ireland. But in Toronto, where F&G went, 18 healthcare workers were affected with considerable mortality and 11 close contacts. So this is just an example of the way these viruses spread. And I want to also add that there was another outbreak in Hong Kong at Amoy Gardens, where the major mode of transmission appeared to be fecal oral. And it had to do with defects in the plumbing in that residence hotel. Next slide, please. So let's turn now to COVID-19 and its epidemiology. So on December 30th, Dr. Li Wenliang reported to his colleagues, most of them medical school classmates, some 7 cases of a SARS-like pneumonia. SARS-like to the extent that antibody tests for SARS were positive. So these would have been the first cases of SARS since 2004. He made the mistake of reporting it to them through social media. And in spite of cautions not to spread the word, the police got a hold of it, interviewed Dr. Li and 8 others in this social media group, required them to sign retractions, accused them of spreading rumors, let them know they were going to jail if they didn't stop. He went back to work, he treated an infected patient, became ill, while many other medical workers were becoming ill. But the irony here is the China CDC was already aware of this epidemic and had been investigating for a week before Dr. Li reported the cases. And they noticed that almost all of the early cases were associated as workers or customers with the Huanan seafood market, one of those live markets again, and they closed it virtually the very next day, January 1st. By January 7th, the Wuhan Institute of Virology had identified a novel coronavirus as the cause. By January 11th, they had shared its sequence with laboratories around the world, and Dr. Li died that day. January 15, the government was still issuing reassurances to the public, which by now had become aware of these strange cases of viral pneumonia. And it wasn't until January 18th when a epidemiologist, a very famous epidemiologist named Zhang Nanshang, was called out of retirement, arrived in Wuhan, and within 2 days he had persuaded Xi Jinping to issue orders to begin the draconian quarantine of Wuhan and then eventually Hubei province. And that day, 14 medical workers caring for a single patient with the disease had become sick. Next slide, please. So this sort of reproduces for you that travel diagram that I showed you for SARS. These are flights from Wuhan, and notice between January 1st, January 25th, there were 500,000 to more than 1 million travelers every day out of Wuhan, all over China. And then it was by January 25th that all travel was shut down. Next slide, please. This is the epidemic curve. You can see that in China, by early March, the epidemic was over, and you can see it was followed by global spread, represented by all of those yellow bars. And they've shown you the inverse response of our stock market. So this is kind of a Trumpian display. If you don't like the way the curve is going, you just turn it upside down. Next slide, please. So how is this transmitted? And so this epidemic has really required us to awake from our slumber about the distinction that we have always made in infection control in the hospital between droplet transmission and airborne transmission. Now, airborne transmission always seemed confusing to me because if you're within 3 feet of the patient and can be infected by a droplet, that seems airborne, but that's not what this means. By airborne transmission, we mean transmission by particles so small that they can remain suspended in the air and be carried by convection currents substantial distances. So conventionally, droplets are more than 100 microns. They fall to the ground within 3 feet. Their size is affected by temperature and humidity. They only get deposited in the upper airway, but they can desiccate to aerosol size. And if you look at this, you can see there's a little problem here. That is, what about particles that are less than 100 microns, but not 5 to 20 microns? In fact, are bioaerosols 5 microns or 20 microns? Obviously, there is a continuum between those two groups that is not accounted for in this system. But those smaller particles, or bioaerosols, can travel in the air. We prevent their spread in the hospital by using negative pressure rooms so that air flows only from the outside, from the hallway, into the room and not the other way. These things are so small, they can reach all the way into the alveoli of the lung. So they're deposited in the lower respiratory tract. Next slide, please. And this gives you an idea of the quantity of the SARS CoV-2 in these aerosols. And you can see that in the case of SARS, which are the blue points on that left-hand slide, it starts well above 10,000 tissue culture infective dose 50s per liter of air. So SARS-CoV-2, a little bit lower, but actually their half-life is about the same as shown on the right-hand slide. But you can see they both can linger in the air for as long as 3 hours. So this is a mode of transmission that worries us a great deal. And these concerns about the difference between droplet and aerosol and the droplets or the particle size in between is what's caused the CDC to extend the recommendation from 3 feet to 6 feet. Next slide, please. This also shows exhaled bioaerosols from 11 different people, and you can see there are 5 of them whose production is negligible, and 6 of them which are quite substantial. So those are the sort of super producers. They may also be the super spreaders that we've been worrying about, and I think that number 10 there with the tallest bar is one of Rachel's friends. But next slide, please. Okay, anyway, so what generates aerosols? Well, singing and talking can, but of course, what the slide I just showed you was of talking. But coughing and sneezing generates a great deal more bioaerosol, singing more than talking. In addition, multiple medical procedures do the same thing. So intubation, even extubation, the COVID-19 test itself, since that also often induces sneezing or coughing, and even non-invasive ventilation generates aerosols. Now, what you see in the picture there is what's called an intubation box, which has just been developed for this epidemic, and only in the last few months, and it's intended to protect that anesthesiologist as he intubates that patient. Next slide, please. So how long does this virus get shed by people who are infected? And it can be up to 6 weeks, but one of the Chinese studies showed very clearly that there is a difference between those who shed it for less than 24 days and those who shed it for more than 24 days. Being male, being delayed in getting into the hospital after the onset of symptoms, having severe illness, particularly being on the ventilator, being elderly, being hypertensive, were all associated with, and even diabetes, with a more prolonged shedding of the virus. Next slide, please. Now, he and his colleagues reported in April the pairing of 77 infector-infectee pairs. And by comparing the serial intervals between cases to incubation periods, they were able to calculate a couple of things. Number one, that the peak infectiousness was between 2 days before and 1 day after the onset of symptoms. Other studies have shown that 40 to 45% of the transmissions occur in those 2 days prior to onset of symptoms. And it can be as long as 4 days prior to symptoms. The basic reproductive number, or R0, in this study was 2.2 to 2.5. Now, what is the R0? The R0 is the number of effective transmissions to other people from one case. So an R0 of 2.5 means that 2.5 people are infected from each case. Now, the R0 for influenza, for example, is 1.3, and we know how successful transmission of influenza is. If, however, you can get that R0 down through intervention below 1, that allows interruption of transmission. Next slide, please. So what are the things that increase risk of transmission? Well, first of all, hospitals without use of PPE, which is what was true in Wuhan in the early days before the January interventions. And of course, airborne precautions. Skilled nursing facilities, and I must say this is going to force us to take a very hard look at the quality of care in nursing homes across this country, which is highly variable and often very poor from a medical and infection control standpoint. Households, and we'll get back to households. Other closed settings, including cruise ships, detention facilities, homeless shelters, any close proximity and duration of close proximity. Loud talking and singing, we've mentioned. Next slide, please. Well, we've mentioned the possibility of fecal-oral transmission and the role it probably played in SARS in 2003. Well, it turns out this SARS CoV-2 can also be detected in stool, especially, but also in blood, ocular secretions, and semen in lower quantities. Something like 41% of COVID-19 patients Now, these are hospitalized patients, so they're sicker, obviously. So this number may be higher than outside the hospital, but 41% shed the virus in stool in high concentrations. Yet only 12-25% actually have gastrointestinal symptoms. You'll remember the GI tract is one of those places where the ACE2 inhibitor is fairly rich on the surface of certain of the lining cells of the colon and small bowel. Shedding continues in the stool actually longer than in respiratory secretions. That is 22 days versus 18 days, up to 29 or 31 days. Next slide, please. SARS CoV is detectable in the environment, and that's why you've heard about those concerns about packages arriving from China and so forth. By the way, stop worrying about packages from China. But in the surfaces in patients' rooms, viral RNA can be detected, especially on toilets. There is greater shedding from some patients than others. You might be reassured to know that cleaned surfaces test negative. It's also detectable in aerosols in isolation wards and ventilated patient rooms where COVID-19 patients are found. But highest actually in what the Chinese called the toilet rooms, which I guess the British call water closets, or those little much more enclosed rooms where the commode is off patient rooms. It's highest in the ICU, as you might expect, and that's up to 113 virus copies per meter squared. Interestingly, in one of these three studies, they looked at the virus in department stores and found that the quantity of virus in the air was greater than in public areas of the hospital. So I think you might compare our own hospitals to say Walmart. I would say I'm far safer in the hospital because everyone is wearing masks, everyone is paying attention to hand sanitizing, than I am in the grocery store. So stop worrying about me. Anyway, viral RNA can be easily detected in aerosol samples within 4 meters of patients. Next slide, please. So I told you we would talk some more about household contacts, and what contact tracing shows us in these several studies is that the rate of contact positivity is substantially higher in household contacts than in contacts outside the household. And I'll let you look at those numbers for yourself, but explain the one that's so different from all the others, and that's the Korean study of 93 cases and their 1,100 contacts. Those that were outside the household actually were almost all in a call center where people were close together, constantly on the phone talking, and obviously that is that's an exception. Next slide, please. So this is a large Spanish seroprevalence study, some 15,000 Spaniards from all over the country who were tested by antibody testing. And they used a couple of different antibody tests, which were a little bit variable, as you can see there, in terms of their sensitivity. But if a person tested had a confirmed case in their household, the likelihood of being seropositive was 31 to 37%. If they had contact with someone outside the household, another family member or a friend or a coworker, their rate of positivity was only 10 to 14%. So clearly, being in the same household for an extended period of time with a contact is the highest risk. Next slide, please. On the other hand, there are outbreaks associated with social gatherings that obliterate that principle. So here is a choir rehearsal in Skagit County, Washington. There were 61 choir members in attendance. One of those persons was symptomatic with COVID-19. There were 53 subsequent cases, 3 of whom were hospitalized, 2 died. There was an Arkansas church who had services or various other activities on 3 consecutive days in March. There were 2 symptomatic congregants at the time. 92 others attended these events. 35 cases ensued, including 3 deaths. There were 26 additional cases among those 35 cases, contacts, including 1 more death. Altogether, 8 hospitalizations. So, of course, everyone's wondering about the gathering in Houston for the Trump rally, and I will tell you that it looks like there are about 62 cases a day being generated from that gathering over this past week. Now, these are daily cases in Nevada, and it's a little bit hard to see, but that greenish blue dotted line on the right-hand side is when we began to reopen in Nevada, and you can see that the case count rose following, even though we had flattened the curve. Now, for the U.S. curve, during June, you'll remember that Vice President Pence was severely criticized for saying that, quote, "we have flattened the curve." Well, that was not entirely incorrect. It was just misleading. Which curve are you talking about, Vice President Pence? You could say, well, he was talking about the death curve. And you could also ask what he means by "we," but that's another story. So that surge in cases that we have seen throughout June in the United States will inevitably be followed by an upturn in the death cases as well, in spite of the fact that the average age of a case has dropped in the United States. Next slide, please. This is the death curve that we have flattened in Washoe County, but we have not dropped it to 0. There were 4 deaths on Friday. There were 2 deaths yesterday. So we're still in that 2 to 5 deaths per day range, and my guess is that we're going to stay there for a very long time, which is what I mean by the long tail of death. Next slide, please. So you've all seen these reports of epidemic models trying to predict what's going to happen, and this is the basic structure of classic old epidemiologic models of epidemics, and they're called the SAIR model, and that stands for susceptible, exposed, infectious, recovered. In other words, this series of compartments, and so the beta number is the proportion of persons exposed to a susceptible, or among the susceptibles. The sigma number, the proportion of those exposed who become infected and infectious. The gamma number, the proportion of those who recovered. And then there's that dotted line that we don't really know about, that should tell us whether some of those who recover might become susceptible again. We actually don't know that. That's the epsilon number. So we don't know how to plug that into the formula. Next slide, please. So there are 21 different models of this epidemic by various institutions. Obviously, the University of Washington's IHME group is the best known, but there's Imperial College London, there's Columbia, there's UCLA, there's University of Massachusetts, and so on. You can see there's quite a bit of splay in that left-hand forecast among these various 21 models. The CDC has tried to sort of average them out for us in what they call an ensemble, and that's shown on the right-hand side. But you can see that the death accumulation continues upward according to all of them. And the faint lines on either side are the confidence limits in that prediction. And you'll remember the University of Washington model has us over 200,000 deaths by November 1st. Next slide. Now this is the R naught number for COVID-19 in Nevada. RT means the actual R naught at a given time. And you can see how it changes in response to human behavior and interventions and so forth. And you can see we got below 1 in April, but in May started climbing again. And at one point, which is at the end of this curve, we had the highest RT in the country at 1.56. However, we have responded. By we, I mean the governor and the state. Our RT right now is down to 1.09. Obviously, we want to see that get below 1, and I'll show you some reasons in a minute. Next slide. This is the minute. All right. So they have an important implication for our ability to control this epidemic in ways you might not suspect. For example, how many people do we need to immunize once we have a vaccine? That depends on continued public health intervention. If we are relying solely on the vaccine, we are going to have to immunize a far larger proportion of the population in order to stop transmission. And that's basically what this slide is telling us. Next slide, please. So these are the comorbidities that are associated with COVID-19. And you can see that across the board here in Italy and China and New York City, hypertension leads the list. So hypertensive people seem to be more likely to be hospitalized or maybe to acquire SARS CoV-2. And diabetes, it makes the list as well. It was only when we got to New York that we recognized that, oh yeah, obesity is also a major risk factor. Next slide. But what we really want to know is what are the risk factors that contribute to death? And there are some surprises here. First of all, in this very large UK study from Oxford and the London School of Hygiene and Tropical Medicine, we have hazard ratios adjusted for age and sex on the left-hand side and fully adjusted with all the risk factors considered on the right-hand side. And you can see that they set the age decile for the baseline at 50 to 59. So that's a hazard ratio of 1. But you can see at each decade beyond that, the hazard ratio rises so that above age 79, the risk of death among those who acquire COVID-19 is 26 times the risk of death for those between 50 and 59. So age remains a major risk factor. And you can see the ethnic hazard ratios there as well. Both blacks and Asians have a higher risk than whites. Next slide. So what about the comorbidities associated with risk of death? Well, you can see that with increasing body mass index, there's increasing risk of death. But the risk associated with hypertension practically goes away. So hypertensives are more likely to get COVID-19, and if this British study is to be believed, are not more likely to die of COVID-19. That's not true for uncontrolled diabetes, however, or steroid-dependent asthma. Next slide, please. There's another surprise in this group, and that is organ transplants. You can see their hazard ratio is just outstanding. I mean, almost 8-fold risk of death in acquiring COVID-19. And that's in contrast to one of the Chinese studies that seemed to show that people with organ transplants were actually protected from death. And so I think what this represents is the British had a much larger sample and therefore a statistically much more reliable sample. So we have to be worried about organ transplantation also. But in contrast is rheumatoid arthritis and lupus. These are folks who are on immunosuppressive drugs, but different immunosuppressive drugs. And those might be slightly protective. Next slide, please. So now I'm going to switch to the clinical characteristics of the disease itself. And this shows you in the upper panel the survivors, and then the lower panel what the Chinese called the non-survivors, euphemism for those who died. Of course, everybody's got fever and cough, but this is idealized because that's not entirely true. Some people have fever and no cough, some have cough and no fever, and so on. Almost everybody gets a fever by the time they're hospitalized, but if they're not hospitalized, they may never get fever. It's 5 or 6 days later, though, that they actually get short of breath, which is that dark blue line in the middle. And it's a few days after that that they get admitted to the intensive care unit. That's the green line. For those who don't survive, the intubation and going on the ventilator happens at about 2 weeks after hospitalization, or day 15. And that's that sort of burgundy line in the middle there, in the lower panel. The interesting thing is the bottom panel, which shows that the virus is being excreted this entire time. All of those blue boxes across the bottom represent viral shedding. And you can say it goes on, you can see it goes on either until death or until about 3 weeks and a day or so before survivors are discharged from the hospital. Next slide. So that's the typical course. But I want to emphasize that this, for those who are critically ill in the hospital, this is not just a respiratory illness. People don't just die of respiratory failure. It becomes, for the critically ill, not for the people outside the hospital, but for the critically ill, it becomes a much more systemic disease affecting all organs, with kidney failure, with liver injury, and so on. And this is one of the first things that we notice, namely, these people get strokes. And this is the incidence of thromboses in the venous system and arterial system in hospitalized patients with COVID-19. And you can see that by the 14th day, about 35% of people do have thromboses. Next slide, please. Now, the virus can also affect the brain besides causing strokes. And on the left-hand panel, you see a photomicrograph with an immunologic stain of cells that contain the virus. Those are those yellow cells, and if you can't see the yellow cells, there are some arrows there to point you to them. And of course, next to that is unstained brain. In any case, you can see all of those brain cells. There are, in one field there are 6 of them that are positive for the virus. And on the far right-hand side is an electron micrograph, the kind of pictures that my mentor Fred used to take, and sometimes with my help. And you can see those typical coronaviruses attaching to a brain cell right there in front of your eyes. Next slide. So how does the virus get in? Well, here's a devastating complication of COVID-19, acute hemorrhagic necrotizing encephalopathy. So those bright areas where you see the arrows are areas on an MRI where there's both hemorrhage and cell death. That's what we mean by necrosis, is tissue death. Next slide, please. Now, this is a young woman in Milan. Boy, that slide got mixed up, didn't it? Anyway, 25-year-old who complained of severe loss of smell. And you'll remember that as one of the newly recognized symptoms of COVID-19. And in fact, it wasn't recognized until cases began to appear in Europe, especially in Italy, where it was first recognized, but also we're seeing it in New York and in the US. So they just happened to do an MRI of this young woman. And here is the olfactory center. This is the part of the brain to which the olfactory nerves that mediate smell lead. And you can see that there's brain injury shown by those arrows in that area of the brain. So for some people, it's going to spread to other parts of the brain from there. Next slide, please. This, in the center here, are the so-called COVID toes that have been described. On the left-hand side is what we call livedo reticularis. You may not be able to tell, but this is looking down on a thigh, and that sort of lacy network of purple there represent areas of decreased skin perfusion. The COVID toes in the middle also represent areas of impaired circulation due to immune injury to microvessels. This is something that's also seen in lupus, for example. It's also seen in frostbite, but I won't get into that. On the right-hand side is a more severe pattern of skin injury due to loss of skin circulation. And so, next slide, please.
Dr. Murphy, I'm sorry, I just wanted to interrupt just for a moment, just to give you a time check. It's now 10:05.
Okay. I'm only going to talk about a couple of the laboratory abnormalities associated with this because they're insightful. One of the first things that was noted by the Chinese, besides a profound drop in lymphocyte counts in patients, is the rise in D-dimer. And you can see, especially in those who do not survive. And D-dimer is a sort of breakdown product of clots. So it's a sign that there is thrombosis going on, oftentimes in microvessels that are responsible for perfusing organs, like the skin that you saw, for example. But it's also activated by the inflammatory response. This represents that overlap between the inflammatory response and the activation of coagulation. On the right-hand side is IL-6, which was also noted by the Chinese to rise, especially in folks who were not going to survive. And IL-6 is at the heart of what's called the cytokine storm, which is this immense, exaggerated inflammatory response that occurs in critically ill patients with COVID-19. And so we have ways of interrupting that process. Next slide, please. Now this is my Barry Hansen slide. People frequently ask me the question, what about these antibody tests? Should I go get one? The answer is no. But this shows you that there's a vast difference in sensitivity between different methods. The method most likely to be available, which is the point of care method that you could use at home, is the lateral flow immunoassay, and you can see it's very insensitive. So it's not ready for prime time. If you can get a chemiluminescent assay, on the other hand, the sensitivity is 98% and the specificity is 96%. That would be quite acceptable, but that's going to have to be done in a laboratory, and I don't know of a laboratory in Nevada that's doing that assay. Next slide, please. So I do want to talk about drug development. We now have, which we didn't really have in April, several ways to treat our patients with who are hospitalized with COVID-19. And so I think our care has improved. We have remdesivir, for example, under an emergency use authorization, and controlled clinical trials do show that it reduces the duration of illness by about 4 days. We don't have good mortality outcomes yet, but it's looking promising. So we can use remdesivir in those patients. We have data from the UK showing that a steroid called dexamethasone reduces mortality in COVID-19 as well. We also have convalescent plasma, which I'll talk about later. And we now know that we need to anticoagulate these patients, which we didn't know in April. Now, there are two promising oral drugs that might allow us to treat patients before they're hospitalized and maybe keep them out of the hospital. One of them is EIDD 2081. It is also inhibits RNA polymerase, but with a different competing analog of a building block of RNA, namely cytosine, than remdesivir. And it's active against remdesivir-resistant viruses. It's coming out of phase 1, soon to be in phase 2, again, it's orally bioavailable, so could be used in outpatients. The same is true with favipiravir, which was developed by a Japanese company for influenza, but seems to have broad activity against many RNA viruses. And it too is an oral drug, and it's completed phase 2, is moving into phase 3, and it looks promising. Regeneron has a couple of monoclonal antibodies that neutralize the virus. It's moving into phase 2 soon. Synerger has an inhaled interferon beta, which is coming out of phase 2, soon to be in phase 3. And then we have two agents that interfere with the cytokine storm. One we already have available and are already using, and that's tocilizumab. Tocilizumab is used for rheumatoid arthritis. It was already approved. We can get it, although there are shortages here and there in the country for obvious reasons, because it is being used. And it looks like it does shut down cytokine storm. There is another drug that's in development that inhibits cytokine storm as well at a different point in the storm. Next slide, please. The bottom line for this slide is that we don't have evidence that convalescent plasma actually works, although one Chinese study suggests it clears the virus more rapidly, but no practical changes in the outcome. Next slide, please. And I do want to show you vaccines in development. Notice that there are several different vaccine strategies here. One of the most interesting is messenger RNA. It turns out that dendritic cells in the skin will take up the messenger RNA. It's coded for the antigens on the virus that are important in neutralizing the virus. And so that dendritic cells can pass this on to antibody-producing cells and to lymphocytes and so on. And it's possible that the antigens could be produced over a long period of time, maintaining a sustained immunologic protective response. Next slide, please. Now, this is something that discourages one in terms of believing that the United States is going to control this anytime soon. You can see that in North America, we are the worst in terms of adopting mask usage as compared to the rest of the country, or the rest of the globe, pardon me. Next slide, please. So these are a few comments about by ID physicians about how this is going to turn out. Neil Clancy at the University of Pittsburgh thinks we're just going to grow tired of mask wearing and isolation, and that we're going to begin to accept politically 2,000 deaths a day, and it'll be 2 years before herd immunity and mass vaccination can contain the epidemic. The other comments are similar. Next slide, please. And so my comments are there are several possible scenarios. One is we could have an effective vaccine by September, even though Fauci thinks it'll be early next year. We could achieve herd immunity by the end of 2020 and terminate the epidemic. That's probably unlikely. I think we're going to have localized outbreaks requiring sustained public health responses, just as we do now with measles and pertussis, even though we have vaccines against those. I think poor global control due to weak public health infrastructure in the underdeveloped world means repeated reintroductions to our own country. I think we're going to have that long epidemic tail of death due to lack of political will. I think that we may have initial success in 2021, but we may then neglect pandemic preparedness and have future pandemics of this, other bat coronaviruses, and families of other viruses. Next slide, please. And that is the end. And Dan, do we want to take just a couple of questions?
What I would suggest is if you want to take a quick spin down through the chat line and see if there's anything there that you wanted to address in particular.
Well, I noticed there were a lot of questions about ACE2 receptors. And I would say, although they're particularly concentrated in the lungs, that they're also widely distributed in other tissues, especially the gastrointestinal tract, even some brain cells. They are the only way that this virus gets into human cells. I would also point out that in 2015, the Bat woman of China, that is the lead virologist at the Wuhan Institute of Virology, who has studied bat coronaviruses ever since before SARS, Shijin Li, had warned us in 2015 that there were several bat coronaviruses which can infect human cells without any adaptation whatsoever. That is, go directly into human cells. Of course, that was ignored. But that's because they can attach to ACE2. Now, the MERS coronavirus uses an entirely different receptor, just incidentally. Okay, so 23andMe is investigating genetic epidemiology. Infectivity varies by blood type. That's true, and type A seems to be the most susceptible. Sally is happy she's O positive. Let's see, how do I advance this? Sweetie, can you figure out how to move up? There we go. I can't seem to get through the chat box. Dan, can you read some of the questions from the chat box?
Sure, there were questions referring to the interrelationship between the virus and ACE receptors. I don't remember if you addressed that in particular.
I'm sorry, say that again.
Interrelationship between, well, actually here we just had a new one come in. What is the efficacy of the Abbott versus the PCR testing?
Well, when you refer to the Abbott test, that is a test for antibodies, so it's a completely different kind of test. Now we're relying on remember, it's out there because of an emergency use authorization, which means the FDA has not taken a close look at their data, and we are relying entirely on the data from Abbott itself. But they did test almost 1,000 samples taken before November of last year. So those should all be negative, right? And only 4 of those were positive. They're either false positives or they're responses to the endemic coronaviruses that cause the common cold. So the specificity there, the ability to identify a negative test seems to be very good. There is poorer data on sensitivity, but I think they studied 18 people serially from the date of onset of symptoms through their illness. There were a handful who were positive on the 3rd or 4th day, and by the 5th day, all of the 18 had a positive test. So that would lead you to think that probably it's fairly specific, but that's not 1,000 people, and that's not even 100 people. And so it's hard to know, hard to be sure of the sensitivity of the Abbott test. The specificity seems to be pretty good. So that's the Abbott test. That tells you, or should tell you, if it's reliable, whether you have had COVID-19 in the recent past. Now, the other thing we don't know is how long that antibody lasts. And it's with the other coronaviruses, it looks like antibody may go away after 4 to 6 months or a couple of years. We also don't know whether that means loss of protection. It may not, because there is in immunology something called the anamnestic response. That is, as the quantity of antibody drops off, even to very, very low levels or negative, nevertheless, the immune system remembers the antigen and has an exaggerated response when the antigen is, such as a virus, or part of a virus, is reintroduced. So that provides protection, even in the absence of continued antibody. So whether the anamnestic response would protect us in future years from this virus, we don't know, but it may very well do so. So that's the antibody test, and that's completely different from the PCR that's done as a standard test for the detection of active infection. The PCR detects the RNA of the virus. Now, what's wrong with that? Well, early in the course of illness, the presence of viable virus, active infective virus, and the RNA of the virus is very tight. But toward the end of illness, as the ability to detect the RNA persists, the ability to detect viable virus begins to fall off. So there's a little separation then, late in the course of illness, between the presence of viable virus and the detectability of the RNA. So it may be too sensitive at 3 to 6 weeks. So we're always happy to see it go negative, but we don't know that a positive test means patients are still infective. One more question, or what do you think, Dan?
We're at about 10:22. If you can provide maybe a quick answer to the question of the hypertension issue. People are curious about the degree to which hypertension is an issue and the significance of that, and with regard to it, whether it's being controlled or not.
Yeah. So it's important to keep hypertension under control. It may be a factor in susceptibility to infection. The British data that I showed you suggests that it doesn't increase your risk of death, if at all. If so, very little. Keep your high blood pressure under control. Now, early on, there was concern about ACE inhibitors, because ACE inhibitors do what scientists call upregulate ACE2 receptors. If you're taking lisinopril, for example, you're going to see an increase in the number of ACE2 receptors in various tissues, including the heart. And so there was concern that that might be a disadvantage. On the other hand, if the lisinopril occupies those receptors, maybe it's protective. So we at last have a study that shows it makes no difference whatsoever. So in the beginning of the epidemic, I stopped my lisinopril. I don't have high blood pressure. I use it as renal protective because I have diabetes, right? So I figured, I could excrete microalbumin in the urine for a few months and it wouldn't worsen my, renal disease or risk of it. But now I'm back on my lisinopril, and you should be too if you need your lisinopril. I hope that answers the question about hypertension.
With that, Dr. Murphy, I'd like to thank you. We'd all like to extend our thanks to you, and I think this is probably a good time for us to take a break. Our service will begin shortly, and we'd invite everyone to please come back for that. And Philip Moore will be speaking on the interrelationship between climate change and COVID-19.
Good.
Thank you very much, everyone. We hope to see you there.